Hello there. Welcome back to Dr. K. Primer lecture series presenting by Dr. K. That's me. Today we will discuss about plasmids. What are plasmids? Plasmids are covalently closed circular extra chromosomal double-stranded DNA molecule. I'm repeating again. Covalently closed circular extra chromosomal double-stranded DNA molecules are called as plasmids. C. Covalently closed circular extra chromosomal double-stranded DNA molecules are called as plasmids. Not all extra chromosomal DNA molecules are not plasmids, but which are covalently closed circular small double-stranded DNA molecules are called as plasmids. Here you see this is a covalently closed circular single-stranded molecule, but that's not the plasmid. If it is a double-stranded, right? You see double-stranded again. Covalently closed circular extra chromosomal double-stranded DNA molecules are called as plasmids. You see, here this is covalently closed circular double-stranded DNA molecules are called plasmids. So plasmids do have two important or three important features. The first one is extra chromosomal DNA molecules. Means here these plasmid molecules do have their own origin of replication. This is the origin of replication through which it can replicate autonomously, independent replication. For that, it requires the origin of replications. Plasmids possess the origin of replication. And the second important point is that plasmids also involved in the various metabolic activities. They are involved in the antibiotic resistance, antibiotic production, fermentation, aromatic compound degradation, hydrogen sulfide production, so on. So many activities. So the enzymes or proteins which are required for these metabolic activities are, those encoded, the enzymes which are required for this, and the respective enzymes are respective genes of those enzymes are present in the plasmids, right? So they encode a spectrum of genes, they are involved in the variety of metabolic activities, right? So they have origin of replication, and they do possess genes which are responsible for various metabolic activities. And also the third element they have a element which is regulate the copy number. So these are the three important elements which are present in the plasmids, right? As I told you, plasmids are covalently closed circular extra chromosomal double strand DNA molecules, and they have origin of replication for independent replication, have elements required for maintenance of copy number, and the plasmid carry spectrum of genes involved in various metabolic activities such as antibiotic resistance, antibiotic production, degradation of aromatic compounds, and resistance to heavy metal tolerance and induction of plant tumor, etc. Right? Here I have given a few activities, right? The plasmids are present in both lower prokaryotes and lower eukaryotes. They are present in the both prokaryotes and lower eukaryotes. Plasmids are used as a cloning vehicles. Why they are so important in the, because they can carry the your gene of interest into the from one host to other host, where it can be replicated, where it can be expressed, right? So they are transport vehicles. So whatever your gene of interest can be cloned and it can move to the different host. It may be the bacteria, it may be the fungi, it may be the plant, it may be the human cell lines. So they carry your gene of interest into this respective host where they can be replicated or they can be expressed into the protein to produce the recombinant protein, right? And there are no natural plasmids and artificial plasmids. Natural plasmids means they are present in the naturally in the organisms, maybe the E. coli or other bacteria. Artificial means they are constructed for the purpose of genetic engineering, right? So artificial plasmids, best examples are PBR 322, PUC 18, PUC 19, PET, and TA vectors, so on. A series, n number of vectors are available, plasmids are available for cloning and expression of the your gene of interest, right? And some of the natural plasmids are also used for the cloning. They are the PSC 101, called E1, RSF 2124. These are the earlier natural plasmids used for the cloning. So this is all about brief introduction about the plasmids. Hope you like it. If at all you like it, just try to subscribe my channel. That's a Dr. K PREM-PRIMER, and you'll get the regular updates. Bye bye.